Hi, everybody.
Hi, Cavalli. How are you?
Yeah,
Giacomo, sorry.
very well, very well. Everything fine. How about you?
I'm fine. How are you doing? How is Montpellier?
Montpellier is good. Yeah, we kind of struggle still with this pandemics, but yeah. How about you guys actually?
We are fine. It's less devastating here, but yeah, disruption is there in the system. Schools are closed. Kids' schools are opening. The elder ones are going to go, I think, from 18th of this month, and the kids, the primary, uh, is going to open in February. Just few hours ago, they announced.
So for the okay, I see. So for the moment they are closed,
Yeah, yeah,
all
yeah.
these schools. Okay, in universities as well.
Yeah, university is also closed, but our semester is going to start 18th. So online classes will start, but they are going to allow physically on campus from 1st of February as well.
Yeah, yeah, I see.
Yeah.
Yeah, we don't know yet. We have actually primaries and secondary schools open.
Mhm.
It's the universities the problem for the moment. We will start the practical works,
Mhm.
but the courses are still online. So
Yeah.
mm,
So our university already decided that except lab courses, where and that also in biology and chemistry labs, they can be physically doable with certain SOPs, but
mm,
all the rest of the courses they are going to go online.
yeah, I
Yeah,
see. So it's very similar.
yeah,
Yeah, it's very tough for. I don't know how many students you have that are coming outside of the city. You know, is it are
in our
most students
case,
local? Yeah.
I think nearly 50% of our population is from outside the city. They require hostel and all these things.
I see. So yeah, so this is pretty much what we have,
Yeah.
and we have a lot of depressions. I mean, the students are really in bad shape because I mean, they when they were confined, they had to stay at home
Yeah,
really all the
yeah,
time.
yeah, yeah,
That was horrible. So now, okay, they can go out, but now we have a curfew that starts. So we already have it actually from 8 p.m. and then they will move it to 6 p.m. So
yeah.
restaurants are closed anyways, bars are closed, museums are closed, cinemas are closed,
Oh God.
libraries are closed, and now you have a curfew starts at 6, and I don't know. So it's like,
Yeah.
so when you have a family, also you somehow stay home, but okay, at least you're with a family, you can do something. But a lonely student is a bit tough.
Yeah, it
Mm,
is. So in
So
our case, a number of students were coming from far-flung areas of Pakistan where, internet connectivity was an issue. So
yeah, this
our
is true.
university did wonderful. So they sent them devices, the internet devices. Some did not have, could not afford the laptops, so they sent laptops as well. So since last 6 months, our university really did, I would say, they went extra mile. And we
Yeah,
also
yeah,
learned how to teach online. I mean, it was a pain in the in the first 3 months, but last semester then was okay. We just submitted grades 4 days ago, so
yeah.
fall semester was okay, I would say. And now we are
Because
planning for.
because actually, yeah. I mean, teaching online. I don't. I mean, my wife is a professor, so she's teaching genetics and epigenetics, and it's so she was really thinking what to do, and then so finally she kind of does kind of the usual. So she doesn't. She was wondering, should I try to set up these tools with a lot of you can ask questions and then collect answers. But that's a bit tricky. so,
Yeah, yeah.
so especially because she has some classes that she has 20 people, but then other classes she has 300 people. So it's very diverse. And so finally she said, okay, I'm giving my talk, and then what she has is some question sessions separate. So she has just one session where people ask questions. So Q&A.
Yeah. Yeah.
But like to have, in principle, you have all these teaching tools that you can have. You can split rooms. You can, make polls and so on, direct live polls and so on. But come on, it's a hassle. It's very hard to do it well, because you can do it, but then if you waste a lot of time, she tried at the beginning, and she wasted a lot of time, and then people get
I can
bored.
imagine.
They do.
We all went through this. I can imagine what she must have been through. It's importantly when it comes to evaluation,
Yeah.
Peter Becker was also in our seminar, and we had similar conversation. And he said, Tariq, problem was they had these WhatsApp groups, and you give them exam, and we asked them, you have to have your Zoom cameras on. And they were talking about all this in Europe, privacy, blah blah blah. You cannot. Peter said, we said you have to keep your cameras on. We cannot trust you.
No, it's the same. Yeah, this is a big problem that they have to. Yeah, exactly that. Yeah, exams online. I mean, how do you make sure? Anyway,
the class mean went up in the first break in March, April. Last year, we used to teach the first year of BS biology, the introductory biology. And one of the exam we conducted, the class mean went to 80%. It never went in that exam. It was like 60, 65. So it went to 80. And then what we did, my colleague who was teaching the module, he divided them into groups. And he came to know one of the student, he was a spy. He said, already groups are done because the groups you have made, they are visible. So 5 minutes before the exam, he shuffled the groups, and then the mean of the exam came down.
My goodness. Okay.
Yeah, but it's challenging.
But anyway, you even if you keep cameras on, you cannot. They can still watch some stuff online and get some. So always. Yeah. So it's hard to. Anyway, you can. There are ways that you make it too long to look for information. So then, it's still they are still more true. But it's a tough thing. Yeah, I can imagine.
So I also teach in last semester. I taught genetics at undergrad, and at the grad level, genetics and genomics, where there's a lot of epigenetics. So at master's level, it's easy because you give them an exam, which is, more experimental. They have to think of, experimental design. Question is something they cannot, take extra help from others. But undergrad is something where you have to stick to textbooks and certain format of questions.
Well, but for instance, we just had the master. So because I teach, actually, I am not supposed to teach, but we do we have a workshop for master for the second year of masters, and so there we have people that can give talks, and then we have sort of roundtable discussions between the students and the speakers. So it was all online now, but still it went fine. And then so half of the marks are what the students have done, how did they interact with the people and so on. So that's kind of an easy thing. But then we also have a formal part. And this, we were thinking we have the students in presence. So we prepared a paper basically where, we give them a text and some bits of figures, and they have to figure out, what are missing controls, what should they do else, and so on. But, it's a published paper. So that finally we could do it with the students in presence, but we thought if they are changing it last minute that we have to do it virtual, it's going to be a mess, because the paper, you can fetch a paper and then of course it's easier to we give them 3 hours to think, because it's really complex thing. But if they can get the paper out of online, they can still read some responses. So we were lucky it didn't change, because we thought if they change things, sometimes like a few days before.
Mm.
You know, then it's a hard thing to think of another exam quickly. You know? So anyway.
And your labs are going okay, so people
Yeah,
are coming routine?
yeah. So actually, yeah. So we have curfew now starting, but so I'm allowed to give them permission. So if they have to come for work, they can. So it's you know, we have very strict rules in the building, and we have plenty of masks, and we have hydroalcoholic gel everywhere, all over the place, and people are very So for the moment, we avoided. So we had some cases, but we immediately isolated them. We traced isolate, and there was no cluster. But okay, that is for the last. So we actually have been closed for 2 months. 2 months in spring last year. So, but then yeah, everything is okay. And we had a bit of shortage also of chemicals and gloves, and we gave a lot of stuff to the hospital as well. We gave all our masks, gloves, and so on to run testing. And so at some point we had shortages, but we never really got to totally tangled. We always managed to do. So we were slowed down, but I would say now it's 100% actually. It's just unpleasant. We have to really do like we have to. First, the lab meetings have been online all the time. So, and usually it was beautiful, the lab meeting, because we took breakfast together, and then we had the lab meeting. It was very nice atmosphere, And now it's all like Zoom, and it's not so nice. Anyway, how about you guys in the lab?
So unfortunately, our labs they closed our labs for 6 months after the first wave. I got permission for 2 people only to come and flip flies, but no work, no experimental work.
That was the same for us.
So for 6 months, closure of labs, that really destroyed everything.
That's really bad. Yeah, for PhD students, it's terrible.
Yeah. And then it's extreme heat here. Summer is very hot, so we had some freezers, things which went off. So quite a big devastation in terms of research, I would say. So you can imagine, if you close 6 months and you have 3 PhDs, each losing 6 months as a PI, you are losing, I don't know, 2 years maybe.
Yeah.
So big
disruption. And so that we had, and we calculated a bit the damage that was made, and so we think it's 2 months closure, but 4 months damage for each person. Because really, I mean, we have some work with the cells. We had to dump all the cells, and some of them were expanded because we had to do biochemistry, all gone. And that by the time you grow it again, it takes a lot of time. And the flies, but I mean, we had some experiments, like transgenerational work we were keeping for many generations. That all dumped, and this is even more than, it's not 2 months. This is more like 6 months.
Yeah.
And but in average, we count about 4 months. So it's, I mean, if you close down 6, it's really tough. And I mean, also mentally it's tough for the people to come back to speed after. So really tricky. Yeah, this is bad for the young people. It's really, and then we asked the university, and that's something that also the government didn't understand. That they did not. They thought they issued a plan to stimulate research, but more in the long run. They say, okay, now every year we're gonna push more money and so on. But that doesn't help the people who got damaged, and they didn't give them the extension that they should have given a half-year extension to everybody.
Yeah.
And didn't happen. So I mean, this is really tough. So that a lot of people have been disappointed. I mean, luckily for our lab, we are okay because we get some funding. So I told you, guys, I take from my grants and I will pay the salaries, and then we will have to make a bit tight with the spending, but everybody gets funded for the time they need.
Good.
But a lot of labs, they cannot do it. If you are already tight you cannot create the money. So
yeah.
it's competitive, you know, so it's very tough.
So your seminar is really going global today, because although we broadcast on Facebook, but we have within Zoom today Professor Qi Zhou from China. He told me, and also from, I just got email from Roberto. He's in Israel. I sent him the Zoom link as well, but he said his net is not working stable. So there are a number of people interested in your seminar. So I think it's time. Let me introduce.
Is it okay if I speak around 50 minutes? Is that something that you, because I cannot shorten it if you want otherwise.
Yeah. So the seminar is standard, like invited seminar we used to have, 50 to 60 minutes.
Okay.
And then, and we try not to disrupt in the middle, because Zoom is a very different media. So we ask questions at the end. And Abdullah also catches up questions from Facebook people if there are questions. And then we have, time at the end for questions.
Wonderful. Okay. Yeah, go ahead when you are.
So let me introduce Giacomo. It's a real pleasure for me indeed to have Giacomo Cavalli today. He studied biology at University of Parma in Italy, and then he moved to ETH Zurich, where he did his PhD with Fritz Thoma and Theo Kohler, understanding the structure and function of the chromatin in yeast. And in 1995, he moved to Heidelberg, to Center for Molecular Biology Heidelberg, as a EMBO postdoctoral fellow in Renato Parro's lab. So this is the pedigree both of us share. In 1999, Giacomo moved to Institute of Human Genetics in Montpellier in France as a junior group leader, and he's still there ever since. Giacomo is actually a real guru in epigenetics. In particular, he has made seminal contributions using fruit fly. He discovered the epigenetic inheritance of new phenotypes that can occur without changes on the DNA sequence. His lab has also discovered that the three-dimensional organization of the chromosome in the nucleus is actually a heritable trait that plays an important role in gene regulatory mechanism. Then Giacomo's lab also identified three-dimensional structural chromosome domains, which are famously known as TADs, for topologically associated domains. And one of many, his lab has also shown that the polycomb group proteins, they behave as tumor suppressors in flies. And Giacomo has, very impressive citations, publications. He's highly recognized and rewarded. He received many awards and distinctions, including EMBO membership, the CNRS, which is the French organization, silver medal, the Allianz Foundation Prize, the Grand Prix 2020, Foundation pour la, this is the French, so I may not pronounce, and two advanced grants from European Research Council. These are really competitive and highly prestigious things. He also served as director for Institute of Human Genetics and Genome Dynamics Department from 2007 to 2010, and also the Institute Director from 2011 to 2014. Over to you, Giacomo. We look forward to hear about the 3D organization of the genome.
Thank you very much. Thank you, Mohammed. It's a great pleasure to be with you today. And let me start maybe sharing my screen, if that's possible. Yeah, looks like it's going. So you should be seeing my screen now.
Yeah.
And all right, so let's start. I will maybe start. I assume that the audience is a little bit heterogeneous, and so I will start with a little bit of introduction. So for those who are experts, bear with me, and I will be boring for the first few minutes, but try to show a little bit what people do to study 3D genome organization and the players that are involved. And then I will move more into the lab research. So this is like a typical kind of picture taken by electron microscopy, looking at the human cell, and this is the nucleus inside. And so you see that chromatin can be seen as a sort of, more or less stained material. The denser material here is heterochromatin, while the less dense material is called euchromatin. And this actually is something that was observed already, more than a century ago by using the standard light microscopes with DNA stains. And what we learned over the years is that heterochromatin is more condensed, it's gene poor, there are repressed genes mainly, and there are some inactive epigenetic marks like DNA is methylated more extensively, and histones have certain marks like primethylation of lysine 9 or histone 3, for example. Euchromatin is more open, it's enriched, genes are more active, and then there are typical active epigenetic marks like histone acetylation or methylation of lysine 4 histone 3. But in euchromatin, you also find repressive genes, and polycomb proteins are actually involved in this repression. And actually, these proteins that can repress genes in an inducible way, sometimes they can lead these genes to really become heterochromatic, and so one can speak of facultative heterochromatin. So these are things that we learned over the decades with multiple methods. But how is actually the DNA folded in chromatin in the nucleus? And this is really a long-standing problem, since if you think in our genome, and of course most of you will know that DNA is a really long polymer. So if we would take all chromosomes, the DNA of every chromosome of a human cell, and stretch it one chromosome after the other, you would reach up to 2 meters. And each chromosome has several centimeters in length. But this actually has to fit in a small nucleus of an average diameter of about 10 micrometers. And so how is it accomplished? So one thing that happens is that DNA is wrapped around histones to form nucleosomes. And this is really a universal feature of chromatin folding of all eukaryotic cells. And then these nucleosomes, but this kind of wrapping of DNA is only compacting about 6 to 10 fold. So you need to compact way more, more than 1,000-fold to put the DNA in the nucleus. So how are nucleosomes then further folded is actually still a mystery, because we know that they form sort of fibers, but what these fibers are has been heavily debated already since the 70s, where people started to identify something that they call the 30 nanometer fiber. But then, whether this fiber exists in the cells has been difficult to demonstrate. And actually, all evidence suggests that there is not a thing like a regular 30 nanometer fiber stretching in the cell nucleus. Rather, maybe a more irregular fiber. So this is already difficult. And then even a 30 nanometer fiber would have only a 30, 40-fold compaction, which is by far not enough. So somehow this has to fall further in three-dimensional space. And this was really totally unknown, such that in textbooks you typically see these pictures like a spaghetti bowl, which really tells us how little we know. And this is something that really excited a lot of labs, and we learned a lot in the last couple of decades about this folding. There are essentially two classes of methods that have been used to really understand in more detail how chromatin is folding. One is relying on imaging, and the other class of methods is more multi-omics. So essentially, it's genomics that is tweaked in a way that you can learn about chromosome conformation. So the imaging, actually, this is a really classical picture from the lab of Thomas Kramer. They have been able, very courageous, actually heroic way to establish DNA fish probes that were specific for each chromosome. It was very difficult because they had to isolate these chromosomes in pulse field electrophoresis, and then isolate the DNA, amplify this DNA. And that is not the end of the story, because if you take this DNA and hybridize it in the nucleus, there's a lot of repetitive sequence in each of the chromosomes. And so this will actually hybridize with all the repetitive sequence in the nucleus and have a horrible background. So to get rid of the background, they had to use a cold, unlabeled, repetitive, enriched DNA that it would compete out all the repetitive hybridization. So that was pretty tough, but they managed to achieve this. And then they used all of the possible different colors in confocal microscopy. Here it's a 7-color, which for people who use confocals is very difficult to get separation of 7 channels. And that's still not enough, because cells have 23 chromosomes. So they astutely combine probes in 2 different colors for some of the chromosomes, such that they could then use spectral reconstruction of the images, and they were able to assign each signal to each chromosome. So in this way, they reconstructed this nucleus where they have each chromosome that has a certain territory. So that's nice. We can learn that chromosomes have specific territories in the nucleus. And so, as that contradicts the spaghetti ball picture that you have seen before. But we don't have resolution. Each chromosome has 1,000 genes, and we don't know where any of these genes is located. So this is something that people have really pushed to try to learn. And there are other methods also that can use imaging that are complementary to what you've seen before. Here, for instance, people have used electron microscopy, and what they do is they deproteinize the chromosomes. This is actually a metaphase chromosome. But now, when you take away most of the proteins, like for instance, you digest with micrococcal nucleus, or you get rid of histones, now the DNA is free, but not completely free. There is a proteinaceous structure that precipitates, that is forming the scaffold of the chromosome, and DNA is emanating out of this structure. And if you would be patient enough to take each of these threads, I don't know if you see them, because I don't know if you have the resolution in your screens, but this gray thing is actually filled with stretches of, with fibers of DNA. And if you would be able to follow each of these, you would see that it starts from the protein structure. It goes out. It is a bit tangled and curvy, and then at some point it starts crawling back in, and it will be ending close to the starting point. So this would be kind of a loop, and people were even able to measure these loops, about 100 of kb or so. And you can see them not just in metaphase, but also in light microscopy and in interface cells. And so you can take nuclei, do the same like micrococcal treatment in permeabilized nuclei. And what you see is that there is a nucleus, but also a halo of DNA signal outside of the nucleus now. And you can measure again this kind of size of the halo. And you can do fish for specific DNA probes. And so you will see that some of them are actually locating outside of the nucleus, so which means that probably this is a part of the DNA that is not retained close to the loop base. And so it's emanating outside, it's in the external part of the loop, but others are actually retained in the nucleus, suggesting that these might be more close to the base of loops. So all very interesting, but this you can do with individual genes, with one gene at a time. It's a very cumbersome kind of analysis. So there were also other things that we were able to learn with microscopy, which is, for instance, that if you try to now preserve the nuclear architecture by using formaldehyde cross-linking and really preserving the 3D structure as much as you can, You can now do FISH inside the nucleus. You can also combine FISH with protein immunostaining. And here you see a typical image of one nucleus of the fly genome, of the fly embryo, where you can stain a protein like polycomb. This is this repressive proteins in green. And then you can stain DNA, and we have two probes, one in magenta and one in red. And these are actually two homeotic genes that are located 10 megabases away, one from the other in the same chromosome. But what we found is that frequently these genes colocalize in the nucleus, which means that they are located in the same compartment. And this is a compartment which is associated with polycomb proteins, suggesting that co-repressed genes can be located in the same polycomb compartment. So distant genes on the linear scale can come closer in 3D, and we were able to show that this kind of property enables more stable silencing of gene expression by polycomb proteins. But again, this is a gene by gene kind of study. Furthermore, we don't have the resolution. We don't know if it is the upstream portion of the gene, the transcribed portion, the downstream portion. We don't have molecular resolution. And this was circumvented by establishing a method, and this was done by Job Decker when he was in Nancy Kleckner's lab in 2002 first, which he called chromosome conformation capture, whereby you generate contacts again by formaldehyde cross-linking, but then you can digest chromatin to make it soluble. And so you get these complexes where you have a contact between two chromatin regions that you can ligate. So you can ligate the DNA, and so you generate a hybrid DNA molecule that never existed in the genome, where two distal portions are joined together, such that you can then study this DNA junction. You can either study it with specific primers if you know what you want to measure. So if you already suspect that you have a contact between two chromatin portions, or you can also use more genome-wide kind of strategies. So 3C, the chromatin conformation capture, is the original method. And then in 2006, people developed 4C, which is one-to-all, which means you take one primer in the gene that you want to study, but then you amplify all of the possible interaction partners of this point, of this gene. And then people developed even further. So the 5C method would be that you now develop thousands of primers in a given region of interest so that you can isolate each possible product between each of the points. So this would be like an all-to-all method, but confined to a certain region of interest. And then IC was the derivative where people, could be able to exploit the full power of high throughput sequencing. And this was, when we could sequence millions or tens or hundreds of millions of reads. So now what you do is just ligate and sequence as much as you can. And actually, if you take large genomes like, already Drosophila and even more so mammalian cells, and you make chromatin fragments like millions, up to 10 million chromatin fragments per genome, each of the fragments can ligate to each of the others. So you can have, an exponential amount of products that can be generated, such that you can really sequence, there's no limit. You can basically sequence even 1 trillion of sequences will still not be enough to fully saturate the information that you have. So for that reason, actually, although IC is probably the most used technique and it's extremely informative, to get more precise information about regions of interest, people develop, again, quite elegant variants of this method. Some of them use protein immunoprecipitation. For instance, if you want to know what are the regions that contact each other depending on a protein like RNA polymerase or CTCF, what you do is you immunoprecipitate the chromatin after ligation, for instance. This is the high-CHI. CHIA-PET is also another variant of this kind of method. Another possibility is that you do a normal regular kind of IC, but then you're going to capture a region of interest. This can be a specific region of a few megabases, or it can be, for instance, all promoters of the gene, so of the genome. So you know what are the contact partners of or the promoters or enhancers or the regions you want to study. And so these are promoter capture or capture IC variants. That's not the end of the story. There's even more. Some people developed methods that allow you not to study bivalent interactions, partner A with partner B, which is typically what you do in IC, but multivalent interactions. There are several flavors of these methodologies. I just list the names here. These are really interesting variations again. And then there are ligation, cross-linking free methods, not ligation free, cross-linking free. So you can either use in vivo microscopy, where you can basically track genes of interest and study whether they contact each other, or you can even use genomic variants. And this is using fusion proteins to the bacterial protein, the methylase. So you can methylate DNA by using these fusion proteins in points of interest. And if you do it in a certain way, you are able to actually track contacts of a given region that you have TAD of the DNA, the DAM methylase, to all of the other regions of the genome. So as you see, we have now an arsenal of methods really. And this enabled people to really study in details what happens in the genome. And this is a typical kind of multi-scale lesson that we learn from applying IC very, very extensively to sequencing through high throughput in the mouse genome. So what we learn is that there are many contacts that are happening in the 100 kb range. So this region is about 400 kb. And you see one of these contacts, and there are thousands and thousands of them. And so these involve two specific points in this region, so that the red signals are strong contacts, and everything in pink is where you have fewer contacts. When you look at what these are in this particular case, these are binding sites for a protein that is CTCF. And actually, if you look at this track, of course, it's very small. I hope that you can see it in the screen. Otherwise, believe me, there are many binding sites for CTCF in this region, but only these two form a contact. And actually, today, I still don't know why the others are not contacting each other. And this is really an open question, a very important open question. CTCF is a famous protein that first was identified as an insulator, kind of protein that can block enhancers from interacting with the promoter. But it's also very important to define these loops, and we see later also to specify boundaries of chromosome domains. But it's not the only protein that induces these loops. There are also enhancer promoter loops. So enhancers can loop out to contact their target promoters. 5' portions of genes are frequently found in contact with the 3' end of the gene. And also polycomb proteins can form regulatory loops. And you will see an example of that later on. If you zoom out now, and so this whole region now would be a small portion of this 8 megabase region that I'm depicting. What you immediately see is that there are all of these triangles that pop out. So there are regions in which you have high frequencies of contact as long as you are in any point within this domain. And then there are, sort of boundaries that separate these domains very sharply. So typically what we see is that they are really following each other. Some regions of the genome they are less defined as domains, so they are kind of interdomain, and then the low amount of contacts between them is probably depending on a specific definition of boundaries that we want to understand. CTCF has been involved in these. Actually, you can see that there are also different kinds of domains, because if we label chromatin with active marks like K36 trimethylation, which is elongation and elongation marker. So where you have a lot of it, you have a lot of active genes. And then you take K27 trimethylation or K9 trimethylation. Here is K27. You see that you have alternations of domains that have more silent chromatin. This is polycomb silencing, and less gene expression, and others that have more gene expression. And typically, these domains, they have separation of histone markers. Now, if you zoom out even more, what you see is that now these kind of domains, and sometimes groups of them actually, form sort of compartments, so that the whole genome, the whole chromosome, is separated in two kinds of compartments. So you either have active portions that will tend to contact other active portions, like you see here. For instance, this guy likes to contact this region that is also active, or this region that is also active, or inactive portions, like these guys that are contacting other inactive portions. So this is called compartments, and it's A or B compartments. And they really separate, compartmentalize the whole genome. So this is kind of, very quick wrap up of what we've learned. And actually, these domains, in particular, what we call topological associating domains, which were these triangles that I've been showing before, seem to really define sort of the regulatory landscape of genes. Because if you take, for instance, a region, this is the mouse genome, and this is coming from the Mundlos lab in Berlin. And you can see one triangle here, then the second triangle here. There is a boundary here, which is actually a cluster of CTCF sites. And actually, you can have genes here. The PAX3 gene, for instance, is a famous gene involved in limb formation. And there are enhancers that are specific of these genes. In the neighboring domain, there are other genes. They have their own enhancers. For instance, the EPH-A4 gene has its own enhancer. Now, this enhancer is activating the gene, but it's actually separated from PAX3. It's not activating PAX3. Now, however, if you have a deletion of a genomic region that includes the boundary, now you have contacts that you can measure here, and you can measure them by 4C or by capture IC. You have contacts between the PAX3 gene and the enhancer of EPH-A4. And that should not happen. And when you have these contacts, PAX3 starts to be activated in the wrong part of the limb, and you have malformations of the limb, of the digits. If you, instead of deleting the whole portion, you have a deletion that encompasses a lot of the intra-TAD region without deleting the boundary, now the PAX3 gene is still separated from the EPH-A4 enhancer, such that it doesn't get hyperactivated and the limb is perfectly normal. Suggesting that really you have a sort of functional separation of genes made by the separation of the genome into different TADs. This has been observed over and over, and so these are all several examples of these, showing that genes within TADs tend to be co-regulated, enhancer promoter contacts are restricted within TADs, and disruption of boundaries leads to ectopic gene expression. But I have to say it is not a universal feature, because in some cases, disruption of TADs has no effect on gene expression. Sometimes there are effects, but they are really small, and people didn't expect them to be so minor. And communications can also occur across TAD boundaries. In some cases, enhancers located in one TAD can activate a gene in another TAD. So this is something, again, that remains to be really understood on a gene-by-gene basis. But there is also another fundamental problem that has been raised. The TADs emerge from doing IC in millions of cells. However, when people try to do IC in single cells, or when they use microscopy to try to correlate, chromatin contacts within or between TADs, sometimes they reported that the separation of TADs is no longer so nicely visible. But on the other hand, single cell IC, for instance, is really a technique that gives you sparse contacts, very few contacts, because every point in a single cell can contact another point or nothing, okay? So you have very few contacts now. So it's very difficult to be sure that you have a TAD or you don't. So we wanted to really study this, and Quentin Szabo and Frédéric Bantigny in our lab dedicated a lot of energy into trying to dissect out this issue. So, and they started with flies, and then they moved to the mammalian cells as well. To study that, they resorted to microscopy, because again, here you can have a single cell readout. Now we can do it to relatively high throughput. And also we have a high resolution that can be provided by super resolution microscopes. And here we use the type of super resolution that we call SIM, structural illumination microscopy. I don't go into the details, but just to tell you that you can get up to 100 nanometer resolution in the actual direction, and in the XY direction, and 200 in the Z axis. So it is already good enough to go close to the size of chromatin domains. And you can perform FISH in a very quantitative way by using oligopanes. So instead of using cloned DNA boxes or last DNA regions that you isolate in pulse field or so, what you can do is design arrays of oligonucleotides that cover the region of interest in a very evenly spaced manners. For instance, you can decide you have 10 oligos per kb or 5 oligos per kb, depending on how large is the region that you want to cover. And this is really powerful, because you know that the signal you get does not depend on how well covered is your region. For instance, if you want to study regions that have a lot of repetitive DNA, you can design oligos in the intervening regions that are unique. And if you want to compare this region to the region that has very little repetitive DNA, well, you cover it in exactly the same way. So you can really compare the two. So when we use this and we develop a probe over 3 megabases of fly genome that is encompassing lots of TADs, what do we see? If you perform the study using conventional wide field microscopy or even confocal microscopy, what you see is sort of, these are the two chromosomes. You see signal. Obviously there is some shape. It's not completely spherical, but what is the shape we don't know. If you do the same in super resolution, you see the picture changes dramatically, and this is a blow up. What you clearly see is you have globules, like this guy, for instance, or this guy, and then between the globules you have some less densely stained regions. You see them very clearly here. But now, just to insist on this point, these dense and less dense regions, the less dense regions are not regions without oligos. Okay, so we have 10 oligos per kb, and so we are pretty much coating the region with oligos. Okay, so if you don't see a signal, is because there is a very low density of DNA in these spaces. So basically, the 3 megabases are folded into globular domains that are then connected by a fine thread of DNA. And so what are these globules then? So what we could do to try to study that in more detail, we developed smaller probes that are encompassing individual TADs, like this probe, for instance, here, or this probe here that corresponds to this TAD. And then we took TADs that were either active, like this one here, okay, that is filled with the K4 trimethylation signal, for instance, or probes that are bound by polycomb, like this one, for instance, that has a lot of polycomb signal. Or in other cases, probes that are located in the chromatin region where you have no gene expression, but there's neither polycomb and neither active marks. Okay, so it's repressed chromatin without any specific marking. And so we can then label each of these probes in a given color, but at the same time we hybridize the global probe that is coating the whole region. So we can have two signals and we can see what is overlaid to what. And this is the result. For instance, take this probe here in a repressive chromatin domain. It's hybridizing a certain region of the chromosome. And if you compare to the signal of the full probe, you see that there is a point of overlap, which is actually one specific globule. If you would do the same with the active chromatin part, which is covering a TAD, you see here the signal is much more scattered and broad. But where is it? It is actually in a region where the global probe has low density of hybridization. If you do a line scan that is crossing through the green probe in a point, you see this signal here. It is in a place where the global probe has little DNA. And what does it tell you? It tells you that the active chromatin is located in a region where there is little density of signal and which is lowly correlated with the general probe, while the repressive chromatin is highly condensed. And more importantly, the repressive chromatin is forming each of these individual globules. Okay, so each time we look at hybridization signals for these repressive probes, they are actually pretty much corresponding to a globule of the total probe. So to be really showing it, demonstrating this precisely, we then devised a strategy where we have probes coating single TADs, and then we take a probe that is coating both TADs, or a probe just overlapping, so that has the same size like the other two, but overlapping the two TADs. So if I'm right, when we take a probe in each single TAD, we should see a single signal, and it should be a globule, okay? But if we take the full or the spanning probe, we should see two globules. And this is what we see in the majority of the case. Single TADs, single globules, spanning probes, two globules. A majority, but not always, okay? So this is actually the single TAD probes. So in some cases, you also have two signals, so they can split up into two signals. And actually for the spanning probe, sometimes we see a single signal for this probe, which means that sometimes you can merge the two TADs. But in more than 2/3 of the cells, each of the two TADs are forming a single signal that doesn't overlap. So if you make a two-color hybridization, the signals don't overlap. And actually, there's less than 10% of volume overlap in 85%, 84% of the cells. So by and large, each TAD is actually forming an individual compartment in the cell nuclei. However, there are violations of this principle. This is now true for Drosophila. What about mammalian cells? In this case, flies have 100 kb per TAD. Mammalian TADs are much larger. They are 800 kb in size on average. And also the mammalian boundaries are mostly located at CTCF cohesin bound sites. Instead, the boundaries between fly TADs can have also CTCF, but many times they have strongly expressed genes that are not bound by CTCF. So the boundaries between TADs differ in the two different organisms. So what do we find in mammalian TADs? So we did kind of a similar experiment, but now, so we look in embryonic stem cells. Now, if you take two probes that are within the same TAD, what do you see? You would expect that because they are in the same TAD, they would overlap. And actually, that's what we see. So the probes are labeled in green and magenta, but there's a lot of white, which is the portion of the signal that is overlapping. And these are multiple signals that we get, where you see, they are sort of interdigitated. This is kind of an average plot between the two. Again, there is overlap between the two. But if we take TAD probes that are labeling different TADs, now we see a clear subdivision of the signals. And you can quantify this very nicely, and you see that there's clear differences for probes within TADs and between TADs. Now, this is not true for just this particular case. We took 15 different regions, and we always see much more overlap for the probes that are within TADs than between TADs, the blue ones. Now, what is very nice is that we can now try to see whether our microscopy data fit with IC, because we have IC for the same cells. And actually, can we calculate overlap for IC? Well, not really. We don't see the signals, but what we can do is we can calculate how many contacts we have between one and the other TAD by looking at the square of the IC signal. Okay, so the signals that are in this square are the intra-TAD contacts, intra-TAD 1, intra-TAD 2, and this is between TADs. So this we can call the crossing contact fraction, which would be maybe corresponding to the overlap fraction in microscopy. And indeed, you see that there's a very strong correlation between the two, suggesting that microscopy and IC are reading the same thing. But the problem with IC, it doesn't tell you what's happening in each cell. So this is very nice. Actually, you can do the same now if you measure the 3D distance. In this case, this is anti-correlate. You know, the more distance you have, the less contacts you have, and this is very nice, verified in the plot. So, what is the role of CTCF and Cohesin in this division of TADs? Because they have been really suggested to be doing so in mammalian cells. And actually, this is how is this happening? People think that Cohesin is an engine that can bind to DNA, and when it binds as a ring, it starts to extrude loops using the energy of ATP, but it is stopped at the edge of TADs by CTCF. And very frequently, actually, we verify that CTCF is binding at both edges of TADs. So if people, when people degraded either Cohesin or CTCF subunits, what they saw is loss of TADs. And this is an example for Cohesin, where you see the TADs are gone. And the same is true also for CTCF. This is a study by the lab of Fejnora, but also others. So, and this is the Rao lab, the Lieberman lab. So, what do we get in our case looking at single cells? We started with CTCF, we inactivated CTCF. And as you see, if you take intra-TAD probes, CTCF shouldn't play a big role. And actually, nothing happens when we take probes within TADs, they overlap in both cases. But the big change is when you take probes between TADs, because they are normally spaced. But if you inactivate CTCF, they completely get tangled, okay, intermingled. And this is true not only for just this region, but in general, such that you completely lose the separation between and within TADs. And why that? Because in every case, what we see is an increase in intermingling of the signals of probes between TADs. What happens now with Cohesin? Well, with Cohesin, actually nothing happens for probes between TADs. So CTCF is still bound, and we know that when you take out Cohesin, CTCF binding is not affected. So actually, the probes between separate very nicely. But the big surprise came when we looked at signal within TADs. Because now what we see is that signals that were overlapping before, they are no longer overlapping at all. And so we're losing overlap. And what this is telling us is that Cohesin normally generates the contacts within TADs. So the fact that you have these intra-TAD contacts really depend on Cohesin. And actually you can see that you can really nicely quantify this. And you can see that this is true for all the regions. So there's again no difference for within and between TADs. But the reason for this lack of difference is the opposite to what we saw in CTCF. It is the loss of intra-TAD contact that generates this kind of equal view of within and between TADs. So the last thing I want to say about this point is that you could conclude, okay, but then flies are kind of similar to mammals. It's just that maybe the role of CTCF is more important in mammals, but like TADs are compartments. This is in part true, but not completely, because when you look at the shape of the signals in mammals, you see that each individual TAD actually has a complex shape. It is not a globular shape, and you see examples of this. And if you take segmentation programs, segmentation softwares, like here is watershed segmentation, you can actually separate the signal into individual kind of nanocompartments or nanodomains, which we called actually chromatin nanodomains. And we could measure the size of these domains, about 200 nanometers in size on average. And you could think, well, maybe could it be dependent on the FISH procedure that we use? We don't think so, because if we take dapistane cells in which we have not done any FISH, we measure the same kind of globules of the same size. And so this is the green signals. So no FISH or FISH doesn't make any difference. And this is a TAD, if you take a probe for the full TAD. So if you measure the size of the full TAD is much larger. So these nanodomains seem to be really like a universal feature of cells. We have measured them in different cell types, and actually they've been measured also in different organisms. Actually, we saw them in Drosophila as well. And what actually drives the formation of these, it is not CTCF and Cohesin, because in the mutant conditions or in the depleted conditions I've shown you before, these nanodomains don't change. The only way we found for the moment to change the nanodomain formation is to modulate histone acetylation levels. If we take ES cells, if we take probe for a TAD, if we treat cells with TSA, you see that the signal gets larger and you can count more of these nanodomains. You can quantify this signal here. So to recapitulate that part, basically TADs are heterogeneous and subdivided into smaller domains called chromatin nanodomains. Within TADs, Cohesin promotes long-range chromatin contacts that might be important for enantiomer-promoter interactions, and CTCF may segregate TADs by preventing Cohesin to cross TAD boundaries. And I want to take the last few minutes to discuss what's inside the TADs. I've been telling you that you have loops that can be formed. This is an example of a loop. Some of the loops depend on CTCF, but others depend on other proteins like polycomb proteins. And so we took this case, the case of polycomb, to really study loop formation and what is its functional role in flies. In flies, you detect loops during embryogenesis. At the very beginning, actually, there's very little structure. In very early embryogenesis, these cells are dividing every 10 minutes or even less. So it is like, super rapid division. Probably they have no time to form TADs. But as soon as cell division slows down a bit, actually, these are syncytial nuclei. It's not even cell, it's nuclear divisions. And so this cycle, the cycle 14 of embryogenesis, you start to have a cycle of 45 minutes. You start to form traces of TADs. And actually, here you already detect this kind of pinpoint, which is a loop. This is then reinforced in later embryogenesis, and you can find many of these in other places of the genome. And very frequently, we found that these loops correspond to peaks of K27 trimethylation and polycomb binding. So we are very intrigued by these. This is an example of a locus, the Dachshund locus, which has two main binding sites of polycomb proteins that are also peaks of K27 trimethylation. And actually, if you make transgenes that contain each of these binding sites, and then you hook to these transgenes a reporter gene, the white mini gene that is forming red-eye pigment, the pigmentation of the eyes is very low, suggesting that you have silencing, and this is polycomb-dependent silencing, because if you measure K27 trimethylation, you have high levels of this mark in these transgenes. So suggesting that these are necessary and sufficient to attract polycomb proteins. Now, if you look in the endogenous region, what we can do now, thanks to CRISPR, we can delete each of these or even both of them together. And we can look what happens after the deletion. So we obtain these lines and we delete each of the binding sites, and we looked at the expression of Daxon. And actually, we were quite surprised at the beginning when taking embryos at different stages of embryogenesis. You know, this is the wild type, this is the normal expression pattern of Daxon. At the beginning, there's actually no or very low expression, and then you start to express it in specific cells, groups of cells, but then you express it in the whole ventral nerve cord. And actually, when you delete PREs, so these polycombining sites, there's little happening. But this is a very interesting feature for us, nevertheless, because it gives us a time where we can actually look at what happens to chromatin without changes in gene expression. So if we see changes in chromatin folding, they will not depend on the fact that you are expressing the gene, okay? So there's no POL2 increase. So that's very nice. And so just to show you that when you delete these polycombining sites, you do have effects, because if you look at K27 trimethylation and you delete just this binding site, actually you see a decrease in K27 trimethylation in the whole region. And this is true also for polycombining. And actually, you will see that this has phenotypic effects later. So it's not an inert PRE, it has a biological function. Now, what happens to chromatin folding? And we can study that by doing fish microscopy, high resolution fish microscopy, where we develop probes against, each of the binding sites here. These are 60 kb about spaced. And normally in a wild type configuration, they frequently contact each other, these two PREs. But if we delete one of them, actually the 2 points start to decrease the frequency of colocalization. And this is true both for deletion of the upstream or the downstream PRE. So you basically have a loop that you can really disrupt. And what is the effect of this disruption? Actually, at the beginning, as I showed you, nothing, but then later on in development, you overexpress Daxon. And the typical sign of Daxon overexpression is actually the generation of this extra-sex comb, which we could see. And so basically, you have a functional consequence for a disruption of a loop. But now you could say, well, you disrupted the loop, but you also take away a polycombining site. How do you know that it is really the loop? So to prove that, definitely, we inserted an insulator site between the two PREs. And this is a very interesting tool, because when we do that, to insert this insulator, we can show that polycombining is not affected. This is actually polymeiotic, it's a PRC1 component. And K27 is also by and large identical to the situation without the insulator. So in cis, chromatin is the same, okay? What happens to the loop? It's actually disrupted because you have an increase in the microscopy distance between the two probes. What happens functionally? And actually we see a gain of function phenotype. However, you can only see it in a small fraction of the population, suggesting that the cis regulatory elements exert, the most of the regulatory function, although you have a small contribution, small but sizable contribution by the loop itself. And the way we interpret that is that polycombinated loops can play an instructive role in silencing. Now, this is repressive loops, but I'm going to show you now that there's not the only kind of loops that you can have. And actually, in the introduction, Mohammed told that how we've shown that polycomb is a tumor suppressor, and so this is a typical example. You can delete a PRC1 protein, you can get these tumors in the eye disc tissue, and a few survivor flies, only 10% survive, have this abnormalized. And we actually were able to show that this depends on signaling. You can have metastases, and then actually if you block NOT signaling or JAK-STAT signaling, you reduce the tumors. And we were also showing that these tumors depend exquisitely by PRC1, because PRC1 mutations generate tumors, but if you mutate PRC2, actually there's very few, and you can even have most of the tissues that reduce their size. So it's a PRC1 specific function. And the reason why that happens is that PRC1, in addition to the canonical binding to regions that have high levels of K27 trimethylation, it also strongly binds to regions that have a sort of enhancer marks. You know, they have K4 monomethylation, K27 acetylation, little levels of K27 trimethyl, and then there's even other regions that are bound that are promoters, and these are active promoters. So that was kind of, you know, something that people didn't really expect. You have polycomb binding massively, actually, to 1,000, more than 1,000 regions that are either enhancers or promoters. So would that mean that we might have some loops between enhancers and promoters that are bound by polycomb? And so to learn that, we did IC in fly tissues, like notably in the eye disc. And actually, what we saw is that if we look at regions that have increasing amounts of polycomb bound to them and are located either in promoters or in enhancers, and we then quantify the amount of contacts between them, the contacts are highest in the regions that have most polycomb bound to it. And so, and we are talking here about active promoters and enhancers. And what are the genes that are underlying that? Well, actually, many of them in gene ontology are enriched in cell-fate commitment, compound eye development, eye morphogenesis. All the big shots of eye development are actually included in this category. So perhaps, you know, this could suggest that polycomb can sometimes assist also activation. And actually, if we look in our mutants, we see overexpression of the repressed genes like the Hox genes that we expected, but we also see repression of these genes that have developmental enhancers that I've been just showing before. And this is a PRC1 phenomenon because we don't see that when we inactivate PRC2 components. So this is suggesting that we can have repressive polycomb loops, but also maybe activating polycomb loops, contrary to expectations. So also suggesting that polycomb has a role in cellular memory of silencing, but also dampening or modulating the expression of genes via active gene loops. And so this is the end of the talk. Thank you very much for your attention. And this is the collaborators that have had in many of our projects and the funding agencies. So thank you very much again.
Thank you. Thank you, Giacomo, for a wonderful talk and a very nice intro about TADs and the methodology to investigate the three-dimensional organization of the, I think I got a whole overview. So questions? Should I maybe stop sharing if you, I don't know. Yeah, you can stop sharing. So questions, let's start questions. Let me see, there was on Facebook also somebody wrote. I'll ask that later on. Grad students, let's start with grad students.
Hi, so I have a question that I was so amazed by the work, and also I'm amazed at how nature has beautifully designed the chromatin structure, that it doesn't allow misorganization or misexpression of even a single gene, and how beautifully it is regulated, that everything is so perfect. So I'm in a state of awe up till now. So, but so my question was that by looking at the TADs that you mentioned in your talk, so the chromatin looping is forming one TAD, what I understood. So how come that the loop or the TAD structure is containing only the repressed genes or the active genes at a time? Because that's what I get from the talk, that one TAD will either be activated form or in the repressed form. So how is it that there is no other gene which needs to be expressed is located in the repressed state of the TAD?
Yes, yes, this is a very, very good question actually. And yeah, I've been probably oversimplistic in my description. So it is true that there are co-regulated genes in TADs. You know, TADs, they can contain, they typically don't contain many genes. So if we look at the size, the average size of fly TADs is about 100 kb. So it would suggest that you have 10 genes per TAD. But actually, what we have is that the more inactive genes that are contained in inactive TADs, they tend to be also larger. For instance, polycomb, the Hox genes, you have only 3 in the inactive TAD of the bithorax complex. So it's not so many genes per TAD, in fact, and also it is possible to have some degree of heterogeneity of expression. What we showed, if you look at the inner structure of TADs, you can take, you can have genes that are silent and others that are expressed to some extent. So if they are very expressed, very strongly expressed, that would actually typically generate a TAD boundary itself, okay? And so, but if you are moderately expressed, actually this is compatible with the presence of the gene within the TAD. However, if you look very specifically at the promoter of this gene, you see a sort of an increased kind of insulation. So actually, at the level of this promoter, you will be isolated partially to what happens, from in the silent portion of the TAD, and you will have more contacts with the rest of the gene that is transcribed. But it is a subliminal kind of effect. So it's not enough for you to be able to see a new TAD forming. You just see an inner structure, okay, an internal structure. So it is actually, there's a lot of fine-grained modulation of these contacts. So as a matter of fact, you have internal structure, you have a lot of modulation of the things, and these are the properties that enable actually a little bit of gene-specific organization within TADs.
Yeah, so the one follow-up question is that these TADs must be dynamic and they must not be static structures in order to have a shift in gene expression from one developmental time to the other instantly. So they
Absolutely.
must be regulated that way. So what do you think would be the driving force or driving protein that
Yeah.
may allow these switches?
Absolutely, very good point again. So, for instance, in the ESS that I've shown you, we can differentiate them into neural precursor cells or into neurons, and we clearly see that there are, in some cases, for instance, you have a large TAD, and then in the NPCs you have 2 TADs that are generated, and at the boundary. And all of these boundaries are very similar. You typically have a gene that has been activated. And then you also have the reverse. Some TADs, where you have 2 TADs, they merge into a single one, okay? And in this case, it's because there was a gene that was strongly active that has been silenced. Now, what we don't know is whether you first have the new structure and then you have the transcriptional response, because we not perform yet a very fine time course of activation or of silencing, and that I think is something that needs to be done really. And yeah, it's a fascinating question, because you could envisage, for instance, when you want to activate a gene, you could envisage that you need to recruit the transcriptional activator and start transcription, and only then you will have a structural separation from the upstream portion of the TAD. Or you could think that first you have, for instance, CTCF that goes there and binds, and you separate the two TADs, and then you recruit the transcription factor to start transcription. And this is something that for the moment is not known. Very good question, and yeah, totally open.
Yeah, thank you so much. Thank you.
Yeah, we have a question from Jing wants to ask question. Jing, please go ahead. You can switch on your camera. Please talk directly to Giacomo.
Hi, sorry, I can't open my camera, but
No problem.
yeah, thanks for your great talk. So I want to ask, how do you think of the roles of centromere on the 3D chromatin formation? Have you sorry?
The role of
The roles of the centromere, the chromosome centromere on the 3D chromatin formation.
The role of the centromere you were talking.
Yeah, role of centromere on the 3D chromatin formation.
So in Drosophila and also in Arabidopsis and probably in other species, there is a phenomenon that is that in many cells you see a configuration called the rebel configuration. So that, you know, in anaphase, so first you have the chromosomes in metaphase, then they separate in anaphase and they are pulled by the centromeres. And then, you know, they go at the pole like this, and then the new cells are formed. And actually in the new cells, all the centromeres are at one pole of the nucleus, and the arms are stretching towards the other pole. And actually throughout embryogenesis, we see that this is actually maintained. And even if you take very late embryos, you keep this organization, which means that some cells, they divided 10 hours ago and they still have this kind of conformation. Actually, people have evaluated that in order to relax this kind of conformation, you would really need a long life for the cells, okay? So the simple fact that you have centromeres is really critical to separate the two arms. And indeed, in IC, we see a big separation between the two arms. In human cells or in mouse cells, well, mouse actually, they have metacentric chromosomes. So there's a specific thing. But in mouse, what we see is that the centromeres cluster together, because there's the centromere and then there is the pericentric heterochromatin, and this together forms chromocenters, and this is actually the same chromocenter like in flies. So that's also a very important feature, because it sort of forms heterochromatic compartments in the nucleus. In humans, where you have a lot of metacentric chromosomes, this is depending on the chromosome. Some chromosomes have a lot of pericentric heterochromatin, others have less. And so in some cases, you have more separation of the arms than in others. So that is more a chromosome-specific role. But definitely, centromere and the pericentric heterochromatin are very important in organizing globally the chromosome. So with differences that the species.
Thank you.
So Giacomo, I have a question from Facebook, Andrea Sanchi. So question is that cohesin sliding and TADS formation, they are only promoted by ATP, or it could be promoted by the positive supercoiling caused by DNA polymerase during DNA replication. This is one question. So in this case, could a treatment with topoisomerase inhibitors rearrange or fuse the neighboring TADs?
Yeah, I'm not aware whether this has been attempted. We didn't for sure, but it's certainly a very good point. And we depleted the full cohesin. What I think it would be a very cool experiment to do, it would be to co-express in these cells, because we know the ATPase domain. So we could co-express a cohesin that is defective in ATP usage. So that would be a very good experiment to do, to find out whether it's really just the energy dependent loop exclusion that generates this effect. Yeah, very good point.
So Mujahid, please go ahead. You have please switch on your camera if you have, otherwise go ahead. Mujahid, can you hear me? Okay, I can ask question in the meantime. So Giacomo, this nanodomains, that's something is really new and interesting to me. So you said if you inhibit with the TSA, you see more of the nanodomains coming up. I was wondering if it is possible to see in normal nanodomain, in a normal cell, if RNA pol 2 is actually in transcriptional mode, or it's in the pause state.
I see. I don't know, but that's something that we have done. We looked specifically at regions where you have more active genes. So TADs that have a lot of expression, compared to regions that have no gene expression. And what we see is that the nanodomains are smaller in these regions. And we explain this by thinking that you disrupt, basically in the inactive regions you have a lot of nucleosome interactions, and these are more disrupted in the active genes. I don't know what it is, so that's what we really will try to find out in the coming years. Is it just histone contacts, or is also pol 2 involved, for instance? What are the determinants of all of this? I think there's plenty of experiments to do. One thing, however, that I need to really insist on, even if you take the most inactive region, of the genome, and so you have a region that has no gene expression at all, no active marks, nothing, and here you are looking whether it forms a single large globule. It does not. It forms individual nanodomains. They have a bit of a larger size, maybe. They will be maybe 300 nanometers instead of 200. And so they might contain, instead of maybe 100 kb or so, maybe more 200 kb, but they will form. And so what we believe is that the nucleosome contact strength is relatively weak. And at some point, once you have large aggregates that are formed, they necessarily fall apart. So there's something that cracks. So you separate these domains from each other. So there's no way in which you can form, and we've looked even in the pericentric heterochromatin regions. We do not have single very large blobs. So it is an inner property of eukaryotic chromatin that you must find everywhere. And I think it's an interesting property, because actually the size of these domains is very close to the average distant size, the distance from inertials and promoter. It is also very similar to the size of the eukaryotic replicon. So in TADs we have origins of replication that replicate the TAD. They all replicate at the same time within a TAD, but you have multiple origins. And what's the size of them? It's about the size of another domain. So we think that there is something very general about how the genome must function that depends on these interactions.
So Mujahid wrote a question. So his question is, are TADs conserved during development, or they change in case of bivalent region genes?
So so TADs of genes that change expression can change. It depends on the magnitude of the change in expression. What I told you before is if there's a strong, sharp increase or silencing of a gene, you typically have a split or a merger of a TAD. But like when you take, so we've done it carefully by looking at the neuronal differentiation. Other people have done it looking at B cell to macrophage reprogramming. you typically have maybe 20 or 30% of the genome, up to maybe 30 or so, that can change TAD position. But the others actually maintain their boundaries. And so there's a global blueprint of the chromosome that is maintained. And then on top of it, you typically have changes. Okay, so a big question is, okay, but why you have this blueprint? And I think that in part this is dependent on the fact that some genes actually are housekeeping genes. In part, actually, there's a boundary that I haven't spoken about, but in addition to CTCF, in addition to active genes, a third boundary is a sharp generation of heterochromatin domain. So when you have a TAD, whatever, with some expressed genes or some repressed genes, when you then enter a region which is rich in lines, and it is heavily decorated with K9 trimethylation and heterochromatic, that point will be the beginning of a silent, a really heterochromatic TAD. And so, and this doesn't change. This typically is always heterochromatic. So this kind of compartmentalization is kind of fixed in the cells. So that's probably why you have a lot of stability from one cell type to the other, but you also have a certain flexibility. Sorry for the long answer.
So Key has a question. Key, please go ahead.
Hi, can you hear me?
Yeah, you're very well.
I have a question about, so how do you think about the mechanism or the function of interaction between chromosomes, which is normally very rare, rarely observed, in Drosophila, but seems to be more frequent found in, species like human and chicken and mouse. In Thomas Creamer's early cytogenetic paper, he already showed, all the smaller chromosomes of chicken tend to be clustered in the nucleus center. And then naturally we would see more interchromosome interaction between smaller chromosomes.
Yeah.
So I'm just really interested in how do you think about the question?
Yeah. So I mean, there's a whole realm of study that has not been pushed yet too much, which is how important would be very long range interactions, actually even in the same chromosome. It could be like one arm with the other arm, or maybe the two ends of the chromosome, or other chromosomes. Now, of course, the fact that you have two genes in different chromosomes, if they would have to stick together all the time to be co-regulated, it's a tough job, because in particular for cells that divide, it's a tough job, because when they divide, they will tend to be separated by mitosis. Yes, but it does exist. In the mammalian case, I think the brilliant example is olfactory receptor genes, so you have in mouse, you have 1,500 olfactory receptor genes that are located in multiple clusters, in multiple chromosomes. And only one is expressed in every olfactory neuron in the olfactory bulb. Okay, only one gene out of 1,500. Okay, and so what he has been able to show is that the repressed genes, they get actually clustered together in heterochromatin. The active gene, it is selected by enhancers, but the enhancer sometimes it doesn't need to be close to the gene. You can have an enhancer. There are some clusters of enhancer that sometimes can activate a gene on another chromosome, and that can happen. It's very efficient. Of course, we're talking about the differentiated neurons, so they don't have frequent cell divisions. So that's good. But still, they have to maintain this interchromosomal interaction for a long time, and this can. I think really the evidence is very solid. In our case, we have shown that you can have interchromosomal interactions between chromosomes within polycombining sites, and they can have also functional relevance implies. So the thing is that we provided evidence, other labs provided evidence, but it's always like one gene at a time. In IC, the reason why is that when you do IC, for instance, the interchromosomal interactions are a minority. You know, most interactions are seen between close by sites. And so you, as I told you, you need to sequence billions and billions and billions of reads before you start to see, you know, statistically significant contacts. So that's making the study difficult. So I think it's open. We don't know much about it. There's really little known, but it's you know, if you have a phenomenon of interest, I would encourage you to push it because it's an exciting part of biology.
Thank you.
So Giacomo, correct me if I'm wrong. So in flies, we thought, you know, PREs on different chromosomes, they make cluster.
Yeah, yeah. So we've shown it with a kind of a proof of concept system. So where we actually inserted transgenes in different positions of the genome, and we can show that they can actually contact a polycomb binding site in another chromosome. Okay, and we have even shown that silencing depends on the contact. Now, I just I shouldn't say too much because it's not my lab, but I've just seen data from another lab. They have measured contacts over long distances between polycomb sites in a very systematic way. So they took, I don't know, I think 20 different sites. And so in many cases, what they see is that, you know, the contact decreases with the distance, with increasing distance. But actually then they have some outliers. So there are some cases in which they see very strong contact, even if they, you know, they are very far away. But to be honest, even they don't know why. So that's the thing. Again, you need to do systematic studies, and then you need to start and study the details of each of the cases. But I have to say, we are very puzzled by this evidence that we have, because we could show that even like 1 kilobase of DNA is able to induce a contact through different chromosomes. And, you know, like if we do microscopy, we can have up to 20–30% of the cells where the two sequences are in contact. So what is it that gives this exquisite sequence specificity? Because this is 1 kilobase of a polycomb binding site that is, you know, it just has a bunch of target sites for Gaga factor and pleiomeiotic, and that's about it. We cannot see anything else. There's full of sites in the genome that have a similar constitution. How can this thing find this particular target locus with such a specificity, while if you take other sites, you know, it will not go there frequently. It will go near 1 or 2% maximum. So how does it do that? We really don't know.
One last question. Lubna, please go ahead.
Yeah. Hi, thank you for the great talk. So I have a question regarding, as you mentioned in your talk, you have calculated the number of contacts, right? I think so in second last slide. So I was wondering if you also checked the number of contacts in the long range. For example, you mentioned you have calculated the contacts between around 25 kb to 250 kb range, right?
Yes.
So this might be within the TAD. So
Yes.
this is somehow already known in mammals and in Drosophila.
Yeah.
Like inactive TADs have more intra-TAD interactions or have more contacts. So I was wondering if you check in the long range.
Absolutely, we did it. We did it, and we also see them. But I have to say, I don't know why we see them. So we see them, but then when you look at, so what we see is, if we look at long range, we see more contacts than expected. Okay. But I have to tell you, we expect very few, because when we plot what's the decrease in contact frequency with increasing genomic distance, it's quite sharp. Okay, so the more you increase the distance, globally in the IC map, without looking at just polycomb sites, if you take the whole chromatin contact, As long as you as soon as you increase the distance, you decrease the amount of contacts. So compared to this decrease, the genomic decrease, when you take polycomb bound sites, you have more contacts than expected, but it's still few contacts, very few contacts. So I don't think that these very few contacts are critical to regulate a gene that is 5 megabase away. You see, it would be, it would be difficult to explain. Instead of having maybe, one contact every 1 hour and lasting maybe for a minute, now you have maybe 2 of these contacts, okay? Yeah. So I don't think this is really like a regulatory interaction. And it could be more than what we think is that these active genes, they are in active chromatin, and active chromatin likes to interact with each other already. So you have two that might, help this kind of interactions. And we see that actually active promoters, just because they are active, they already contact each other a bit more. And so what I believe is that already this is happening. And then if you have polycomb on top of it, it's helping it even more, okay? Yeah. So what could be is that these kind of contacts maybe give a little kick to transcription, but an individual contact will have very, very little input, a single one. But the fact that you have this global organization, this helps the gene to be expressed, okay? So in the long range, it's more like you're compartmentalizing the genome such that genes, they tend to be in active regions in active compartments of the nucleus, and that helps a bit to express. In the case of short range, it's more like enhancer promoter contact. I mean, it's the specific enhancer of that gene that goes find the promoter. So it's a different level of function, and which is in a way it's also like the silencing loops. I think that the key signal is binding of polycomb close to the promoter. And then you have a second input, which is, well, if you have a loop coming from a distal region, you generate a region of the nucleus that is filled with polycomb proteins. So if you lose the protein from the proximal PRE, well, maybe some other protein is coming from the distal PRE that can substitute for a few seconds. So it's helping to stabilize the silencing.
Yeah, actually I asked because maybe you have also read the work from Peter Becker lab. So actually they have shown in Drosophila embryonic stage, I think so late embryonic stage. So they have shown in the long range interaction, they find some cis regulatory elements, which calls PNOX. So they interact more in the long range. So that's why I thought maybe you have also checked, because in the short range, you check the correlation with K27ME3. Yeah, but thank you. Yeah, makes sense.
That's very good work actually from Peter, absolutely, and very interesting. And so it's a sort of, it's going in the same direction.
Yeah. Thank you so much.
Giacomo, there's a very good question. So one of my grad students is asking if cells have a TAD blueprint, do disease like cancer that drastically change the transcription profile of cells affect that blueprint? And if yes, you ever envision it being used diagnostically?
All right. So now, if you take cancer cells that those that we have in culture, like HeLa cells, and so on, that is devastating. Actually, this is true. The blueprint is gone. Blueprint is gone. So there are many correlates with TADs. For instance, the active genes, they tend to be located more close to the boundary of TADs. Active origins of replication in normal cells tend to be located close to the boundary. You do that, you look in cancer cells. Yeah, you also see some TADs, but they are fully misorganized, and there's no correlation now, the position of a gene or an origin of replication, the timing of replication, they are totally decorrelated now. Okay, so this is when you take these kind of cells, but these are very weird, that with thousands and thousands of mutations of rearrangements, and They have been selected for decades, you know. So if you take a primary cancer, how is the situation? Now we have fewer data, but actually we started to do this work by looking at the patients that have multiple myeloma. That's a very, very severe cancer, unfortunately, for the moment, incurable cancer. And there we have seen that you have very strong changes, but not completely destroyed the blueprint. So the blueprint is still there, but you have many more changes compared to, let's say, a normal cell that is maybe differentiating from another cell. And actually, yeah, this is very interesting because we know that kind of cancer, so we know the kind of rearrangements that are frequently involved, and the blueprint, you know, reprogrammings are typically hitting into genes that are candidates for being oncogenes or tumor suppressors. So that's very, very interesting. And so, I mean, it definitely can have diagnostic value, even maybe prognostic value, because for cancers that have different degrees of severity, probably you can reach that point. Now, I'm not to the point. I mean, we personally, we are not developing like, you know, diagnostic tools or so. It's still an expensive kind of methodology. But yeah, I think you can probably, out of that, you can maybe develop smart assays that can be used. You know, once you know where you are, what maybe you can simplify. Instead of having like high C, what you do is more like a reduced version where you are assaying specific regions that are you know, important for a particular cancer that you want to study.
Good. Thank you. Let's all thank you, Giacomo, for sparing his time generously with us. We are 1.5 hours, or even more. We started 15 minutes earlier than 2 here. Thank you, Giacomo. It was a real pleasure. We wish you a good day there. And we would like to stay connected. And I'm pretty sure we are going to bother you more than often.
Thank you very much. It was great. Had an incredible times with the discussion. Great questions. Thanks a lot. Thanks very much.
Thank you. Ciao.
Bye-bye.
Bye-bye.